Have you ever asked yourself why a cell needs to undergo cell division or cell reproduction? To know the answer, just sit back, relax, and enjoy the discussion because at the end of it, we'd be able to understand chromosome structure and organization in eukaryotic cells, identify the stages of the cell cycle through images and by description of major milestones, identify and explain the important checkpoints that a cell passes through during the cell cycle, identify the stages of mitosis and meiosis by photos and by description of major milestones, explain why meiosis involves two rounds of nuclear division, and lastly, examine karyotypes and identify the effects of significant changes in chromosome number. Cell division is key to life. From the moment we are first conceived, we are continually changing and growing. In order for our bodies to grow and develop, they must produce new cells and allow for the death of old cells. Cell division is also an essential component of injury repair. If our cells couldn't divide and create new cells, our bodies could never produce new skin cells to heal a rash or grow a fingernail back. However, when cell division goes wrong, dramatic results may occur. Without sufficient cellular oversight or supervision, repeated rounds of unregulated cell division can lead to a minor condition like psoriasis or a life threatening disease like cancer. Cell division undergoes a strict cycle with multiple stages and checkpoints to ensure things don't go wrong. Additionally, without cell division, no species would be able to reproduce. Life would simply end. A cell cycle is defined as a series of events that takes place in a cell as it grows and divides. There are three cycles in the cell cycle. We have the chromosome cycle, cytoplasmic cycle, and centrosome cycle. So let's start first with chromosome cycle, wherein DNA synthesis alternates with mitosis or karyokinesis or nuclear division. So in here, DNA synthesis is a double-stranded DNA molecule that is copied to produce two identical DNA molecules. And during mitosis, the duplicated copies of the genome are ultimately separated. So cytoplasmic cycle, cyto as in cell. So cell growth alternates with cytokinesis or cytoplasmic division. During the cell growth, its components also become double in quantity, and during cytokinesis, the cell divides into two. Lastly, centrosome cycle. So this is actually a requirement of the aforementioned cycles for the formation of the two poles of the mitotic spindle. That's why this cycle is very important and is considered the third cycle in the cell cycle. The cell cycle is divided into four phases or stages. We have the G1, the S, the G2, and the M phase. Let's start with the G1 phase. G1 is an intermediate phase occupying the time between the end of cell division in mitosis and the beginning of DNA replication during S phase. During this time, the cell grows in preparation for DNA replication, and certain intracellular components, such as the centrosomes, undergo replication. Before a cell begins DNA replication, it must ensure that it is biologically ready to take on such a process. G1 is the phase when this cellular monitoring takes place. During the G1, the cell reviews the cellular environment and the cell size to ensure that the conditions are appropriate to support DNA replication. Not until the cell is ready does it leave G1. If all is not ready to undergo DNA replication, cells can pause during G1 and enter a phase called G0. Depending on a cell's preparedness to continue in the cycle, G0 can last days, weeks, or even years. When the cell has reached an appropriate size and is in a supportive environment for DNA replication, it will exit either G1 or G0 and enter the next phase of interphase called S phase. S phase or synthesis is the phase of the cell cycle when DNA packaged into chromosomes is replicated. This event is an essential aspect of the cell cycle because replication allows for each cell created by the cell division to have the same genetic makeup. During S phase, a number of events additional to chromosome replication take place. Cell growth continues through S phase, as does the rate of synthesis of a number of proteins and enzymes that are involved in DNA synthesis. Once DNA replication is complete, the cell contains twice its normal number of chromosomes and becomes ready to enter the phase called G2 phase. Similar to G1, G2 is an intermediate phase, a time for the cell to ensure that it is ready to proceed in the cell cycle. Occurring between the end of DNA replication in S phase and the beginning of cell division in mitosis, G2 can be thought of as a safety gap during which a cell can check to make sure that the entirety of its DNA and other intracellular components have been properly duplicated. In addition to acting as a checkpoint along the cell cycle, G2 also represents the cell's final chance before it is split into two independent cells during mitosis. Interphase is what happens before and after mitosis. It is a sort of resting period between replications. While the cell is not doing anything as dramatic as splitting in half, it still does some important things in order to prepare for the stages of mitosis. In fact, cells spend most of their time in interphase. The interphase is characterized by the following features. First, chromosomes occur in the form of diffused, long, coiled, and indistinctly visible chromatin fibers. Second, nucleolus is greatly increased due to the accumulation of ribosomal RNA or rRNA and ribosomal proteins. In animal cells, a daughter pair of centrioles originates near the already existing centriole, which means there are two pairs of centrioles in the interphase. Additionally, the net membrane biosynthesis increases during this stage. Before we move on to the next stage, let us first scrutinize the chromosome structure. Chromatin is a complex of macromolecules composed of DNA, RNA, and protein, which is found inside the nucleus of eukaryotic cells. Chromatin exists in two forms: heterochromatin or condensed, and euchromatin or extended. The primary protein components of chromatin are histones that help to organize DNA into bead-like structures called nucleosomes by providing a base on which the DNA can be wrapped around. A nucleosome consists of 147 base pairs of DNA that is wrapped around a set of 8 histones called an octamer. Linker DNA can be seen extending from one nucleosome to the next. The nucleosome can be further folded to produce the solenoid chromatin fiber. Solenoid chromatin fibers are coiled and condensed to form chromosomes. Chromatin makes it possible for a number of cell processes to occur, including DNA replication, transcription, DNA repair, genetic recombination, and cell division. So here is the bead-like structure called nucleosome, and each nucleosome is connected to another nucleosome by what we call as the linker DNA. So the nucleosome can be further folded to produce this solenoid chromatin fiber, and the solenoid chromatin fibers are coiled and condensed to finally form what we call as the chromosome. Again, chromatin structure is organized at several levels. These are the chromatin primary structure, the chromatin secondary structure, and the chromatin tertiary structure. So the primary structure of chromatin is what we call as the nucleosome. The secondary structure of the chromatin is the solenoid chromatin fiber. And lastly, the tertiary structure of chromatin is the chromosome. Okay, so let's try to test ourselves. So this thing here, this nucleosome is the chromatin primary structure. This one right here is the chromatin secondary structure, or what we call as the solenoid chromatin fiber. And this one right here is the chromatin tertiary structure, or the chromosome. People often confuse these three terms: chromatin, chromosome, and chromatid. While all of those three structures are composed of DNA and proteins within the nucleus, each is uniquely defined. As mentioned, chromatin is composed of DNA and histones that are packaged into thin, stringy fibers. The chromatin undergoes further condensation to form the chromosome. So, the chromatin is a lower order of DNA organization, while chromosomes are the higher order of DNA organization. Chromosomes are single stranded groupings of condensed chromatin. During the cell division processes of mitosis and meiosis, chromosomes replicate to ensure that each new daughter cell receives the correct number of chromosomes. A duplicated chromosome is double stranded and has the familiar X shape. A chromatid is either of the two strands of a replicated chromosome. Chromatids connected by a centromere are called sister chromatids. At the end of cell division, sister chromatids separate and become daughter chromosomes in the newly formed daughter cells. Chromosomes are thread-like structures in which DNA is tightly packaged within the nucleus. DNA is coiled around proteins called histones, which provide the structural support. Chromosomes help ensure that DNA is replicated and distributed appropriately during cell division. Each chromosome has a centromere, which divides the chromosome into two sections: the P, or the short arm, and the Q, or the long arm, where P stands for petit, which means small. The other arm is called Q because it is the letter that follows P in the English alphabet. The centromere is located at the cell's constriction point, which may or may not be the center of the chromosome. During mitosis, spindle fibers attach to the centromere via the kinetochore. At the end of each chromosome is a repetitive nucleotide sequence cap called a telomere. In vertebrates, the telomere is a TTAGGG sequence, repeated to approximately 15,000 base pairs. These DNA regions serve a critical role of preserving the genomic sequence by protecting the genome from degradation. Additionally, it also inhibits chromosomal fusion and recombination. These regions are also involved in chromosome organization within the nucleus. Normal chromosomes each have a single centromere. Its position along the chromosome can vary. Metacentric chromosomes have the centromere located midway between the ends of the chromosome, separating the two arms of the chromosome. Chromosomes with centromeres positioned visibly off center are called the submetacentric. Acrocentric and telocentric chromosomes have centromeres positioned very near and at the end of the chromosome, respectively. In the case of telocentrics, the centromere is at the end of the chromosome adjacent to the telomere. Karyotyping is the process of pairing and ordering all the chromosomes of an organism, thus providing a genome-wide snapshot of an individual's chromosome. Karyotypes are prepared using standardized staining procedures that reveal characteristic structural features for each chromosome. Clinical cytogeneticists analyze human karyotypes to detect genetic changes or anomalies involving several megabases or more of DNA. Karyotypes can reveal changes in chromosome number associated with aneuploid conditions such as trisomy 21 or Down syndrome. Careful analysis of karyotypes can also reveal more subtle structural changes, such as chromosomal deletions, duplications, translocations, or inversions. In fact, as medical genetics become increasingly integrated with clinical medicine, karyotypes are becoming a source of diagnostic information for specific birth defects, genetic disorders, and even cancers. This is the organized profile of a human's chromosomes. Based on size and shape, the chromosomes are classified into 7 groups. Groups A, B, C, D, E, F, and G. Group A, which includes chromosomes 1 to 3, are large metacentric. Group B, which includes 4 and 5, are large submetacentric. Group C, which includes 6 to 12 and chromosome X, are medium submetacentric. Group D, which includes 13 to 15, are medium acrocentric. Group E, which includes 16 to 18, are short submetacentric. Group F, which includes 19 and 20, are short metacentric. And lastly, group G, which includes chromosomes 21 and 22, and Y, are short acrocentric. I have here diagrams of 2 male individuals that have genetic disorders. On the left is a karyotype of a male suffering from Down syndrome or trisomy 21. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Trisomy 21. And on the right is a karyotype of a male suffering from Klinefelter syndrome. Instead of normal XY, this person has double XY. Now that we have finished characterizing chromosomes, let us now continue with the cell cycle. A checkpoint is a stage in the eukaryotic cell cycle at which the cell examines internal and external cues and decides whether or not to move forward with division. There are a number of checkpoints, but the 3 most important ones are the G1 checkpoint at the G1 to S transition, the G2 checkpoint at the G2 to M transition, and the spindle checkpoint or the M checkpoint at the transition from metaphase to anaphase. The G1 checkpoint is the main decision point for a cell. That is, the primary point at which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enters S phase, it becomes irreversibly committed to division. That is, barring unexpected problems such as DNA damage or replication errors. A cell that passes the G1 checkpoint will continue the rest of the way through the cell cycle and produce 2 daughter cells. At the G1, a cell checks whether internal and external conditions are right for division. Here are some of the factors a cell might assess. First, the size. Is the cell large enough to divide? Second, nutrients. Does the cell have enough energy reserves or available nutrients to divide? Third, molecular signals. Is the cell receiving positive cues, such as growth factors from neighbors? And lastly, DNA integrity. Is any of the DNA damaged? These are not the only factors that can affect progression through the G1, and which factors are most important depend on the type of cell. For instance, some cells also need mechanical cues, such as being attached to a supportive network called the extracellular matrix, in order to divide. If a cell doesn't get the go-ahead cues it needs at the G1 checkpoint, it may leave the cell cycle and enter a resting state, which is the G0 phase. Some cells stays permanently in G0 while others resume dividing if conditions improve. To make sure that cell division goes smoothly, the cell has an additional checkpoint before M phase called the G2 checkpoint. At this stage, the cell will check the DNA integrity and the DNA replication. If errors or damage are detected, the cell will pause the G2 checkpoint to allow for repairs. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted, and the cell attempts to either complete DNA replication or repair the damage of DNA. If the damage is irreparable, the cell may undergo apoptosis or programmed cell death. This self destruction mechanism ensures that damaged DNA is not passed on to daughter cells and is important in preventing cancer. The M checkpoint is also known as the spindle checkpoint. Here, the cell examines whether all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of the sister chromatids during anaphase is an irreversible step, the cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite poles of the cell. How does this checkpoint work? It seems that cells don't actually scan the metaphase plate to confirm that all of the chromosomes are there. Instead, they look for straggler chromosomes that are in the wrong place, such as floating around in the cytoplasm. So, if a chromosome is misplaced, the cell will pause mitosis, allowing time for the spindle to capture the stray chromosome.